Now you can
Okay.
switch on your video.
Yeah, now I am, yeah. And now I'm sharing my screen again, okay?
Yes. So good afternoon to one and all. Welcome to the afternoon session of FDP. With the kind consent of the convener, we are starting the afternoon session of this FDP program. Our guest for today is Mrs. Ekta Prasad. She has done her MSc in biotechnology and also holds a postgraduate diploma in intellectual property rights from National Law School, Bangalore. She is a patent agent and a consultant on patentability of the invention and also facilitates in filing patent application and preparing examination report reply. She is associated with KSCST. Ma'am, on behalf of the staff, student and the management of the Oxford College of Engineering, I, Salma, take it a privilege in welcoming you to this session on patenting. Uh, Welcome to the department, ma'am, and welcome to the FDP Phase 2 session.
Thank you, Salma ma'am. I think there's a glitch when I'm sharing. My video is getting off.
That's fine, ma'am. Now you can share your screen and you may
PPT, yeah,
start.
yeah,
Yes.
yeah.
You may kindly start.
So is it in full screen, ma'am?
Yes, ma'am.
Okay. Okay. So good afternoon, everyone. Thank you so much, Oxford University, for providing me opportunity to give a talk on this topic. And thank you, Salma ma'am, for coordinating everything. Okay. So here I'll start. So first, what is a patent? Now, first, when we see patent in biotech, so first we broadly will see where the patents stand in the technology level. So, patent is an exclusive right provided to a person who has come up with any invention or any innovation, and the government or the state gives a reward to the patent holder. Okay, so basically, the patent system rewards the creator or the innovators from whom a product or process is in the stages from his or her creative mind. Okay, so and this patent is validity is for 20 years. Like for 20 years, you can relish the exclusive right to make, sell, use the invention, and to exclude the third party from using your invention. Okay. So, as per TRIPS, Article 27, Para 3B, patent is for all the technology. It penetrates to all the technology. It means in every technology, whoever is the innovator, whoever is the creator in that field, who comes up with a product or a process, and which passes through the patentability criteria, he will get the patent granted and he will enjoy the exclusive rights provided to him. Okay, so same now my topic is broadly focusing on the biotechnology. So patent also penetrates or it goes in the biotechnology field where the system uh, uh, like rewards the innovator and the creator in this field. Okay, so patent system emerges in the beginning to promote industrial innovation. Hello, yeah.
Ma'am, we thought a slide was not changing, so it's okay.
Okay, okay. Okay. So now one of the basic or the main problem with the bio, and my voice is fine. Is it okay?
Yes, ma'am.
Okay. Thank you. Actually, I'm not seeing anyone and I'm talking, so it's a very weird situation. So anyway, we are coping. Okay. So one of the main problem with the biotechnological innovation is the raw material in this technology is the living organism. Okay. So here we are manipulating or we are using the living organism in the from this technology. So that is the main problem. So here in the patent system, we grant patent to invention, not to discovery. So in biotechnology, when the raw material itself is a living thing, it means it is a discovery. It is there in the nature. It is nothing to be invented. So in biotechnology, the examiners or the persons who are checking the biotechnological invention to make it patentable or not, they have to demarcate, or they have to say whether this product or process is an invention or a discovery, because it is a highly chance that it comes into the discovery, because the raw material in biotechnology is living things. Okay, or maybe we are using the processes from the living thing, that is from the microbial process or essential biological process. So we should not take anything from the nature and go for patenting. Okay, because that is not an invention, that is a discovery. So invention in biotechnology means where is the human intervention, where is the human creativity in the raw material which is a living, or which is extracted from nature. Where is the human intervention, where is the human intellect into the raw material which is giving to this product. Okay, so that is the main challenge. Every technology, if you go to computers, then if you go to mechanical, chemical engineering, in all this technology, patents are there, and each technology has their own challenge. So biotechnology, the main challenge is the raw material and the human intervention. We have to make a demarcation, like whether this product or process claiming to be a patent, or seeks a patent, is an invention or a discovery. Okay, so in all the technology, as per TRIP, it has to pass through certain filters for any product or a process to be patentable. Okay, so this is common to all the technology, that is novelty, inventive step, and industrial applicability. The same applies to the biotechnology. So here also novelty, inventive step, and industrial applicability. So what is novelty? So novelty basically means which your product or process, when you're coming for protecting it, it should be novel. It means it should be not there in the prior art. Prior to your application, there should not be present in the prior, before it. It can be either in the form of publication, either in the form of use, or either in the form of any knowledge, even in the journals. Okay. Then next is inventive step. This step is very important in biotechnology, because here it decides whether the invention is obvious or not. Because what happens, like many innovators come with an invention which is very obvious. If a person skilled in the art, or if a person is from that technology, if he sees and if he analyzes it, he or she will say it is very obvious. If a person know ABC of this technology, it is very obvious to come up with this invention. So your invention should not be very obvious. Okay, it should have a creative light into it to be patentable. Next is industrial applicability. So you cannot come up with anything without any industrial application. You cannot come up with any gene and say, okay, this is the gene sequence, but you don't have any function of it. Then it won't be patentable. Okay, so these are the major three things for an invention to be patentable: that it has to be novel, it has to have an inventive step, and it has to have an industrial applicability. Okay, and specific for biotech inventions, there are certain exclusions. Okay, so if we see the Patent Act 1970, Section 3, there are list of exclusion which surpasses to the different technology. There are list of exclusion which is which says that these subject matters are not patentable. Okay, so these are section B, C, D, E, H, J. So I will during the course of my lecture, I'll discuss this. Okay, so first is novelty. So novelty, as per section 21J of Patent Act, says that invention means a new product or a process. It has to be new, okay, involving an inventive step and capable of industrial application. So what I told, all the 3 points, all the 3 things should be there, but newness here means novelty, and newness means that invention is patentable only if in the light of prior art it is not anticipated, means it is not present in any form prior to your application or prior to your claiming for an invention to be patentable. So for example, a claim to a product, if like if any sequence, if any DNA sequence is known and it is there in the database that this sequence is known, but you come up with a new process to make the same sequence, then you will not be granting a patent, because you are coming with a product which is already known, but you are not claiming the product, you are claiming the process. Okay, so the claim to a product obtained or produced by a process is anticipated by a prior art disclosure that particular product per se, regardless of the method. Means whatever method you come up with that product which is already known in the prior art, that method won't be patentable. That method will be patentable only if you are coming up with a product which has a distinct property or which has some enhancement, which has some special property which is not known before. Okay, so here, so if any you are coming up with a process, claiming a process to be patented for a product in which the product is already known, then that process will not be eligible for patent. That process will be eligible for patenting only and if the product what you are coming up has an enhanced property from the prior art. Okay, so example is if you're a polypeptide or a compound which is a product of a method, okay, which is a product of a method claimed by X, like if you are claiming for a method to get a polypeptide or a compound, and that polypeptide and a compound is already known, then that method will not be eligible for patenting. Okay, only that process will be eligible for patenting if your product will have an enhanced or a distinct properties. Okay, so there only your innovation comes, or as a human intervention. Like in polypeptide sequence, like claim to a polypeptide sequence which is cannot be given if that polypeptide sequence is already there in the database. Okay, this is due to novelty, and sometimes what happens, it is not always true that if two sequence are same, it means it will give the same function. Okay, so the polypeptide should not be there in the prior art. If it is there in the prior art, then it won't be getting a patent. Next is your inventive step. So here, anything which is obvious, if you create certain mutations in a sequence and you come up and claim a patent for it, then, and if that mutation is nonsense, if it is not giving any good protein, or if it is not giving any sense protein, then that will be away from patenting arena. Okay, so suppose I'll just give an example. A recombinant DNA sequence encoding a human interferon polypeptide is the claim. In the prior art, the sequence of alpha 1 is already there. Okay, so here the inventor is, or the person is claiming for interferon alpha 2 polypeptide, and in the prior art alpha 1 polypeptide is already there, and there is lot of homology between alpha 1 and alpha 2. Okay, but patent was granted to him because the alpha 2, the claimed polypeptide, has an enhanced property, okay, 30 times more potent for a viral activity. Actually, this human interferon alpha 2 polypeptide was claimed for the antiviral activity, and this polypeptide had enhanced like 30 times more potent in the antiviral activity. So that's why, because here it is the enhancement. Otherwise, in the prior art, already human interferon alpha 1 was known. But here enhancement is its property, or it is in its functionality. Next is the utility or industrial application. So any invention, you cannot just go with any protein peptide receptor claiming that this is the product, but without any application. Okay, so every invention should have a substantial, specific, and credible applicability or utility, or it should have an industrial applicability, and the subject matter should have this utility part. Okay, so I'll give an example. Here, one innovator came with a V28 protein, which is nothing but as a receptor sequence, a polypeptide sequence, and which has had much homology with 7TM receptor. And the functionality of V28 was not very clear. So in the application, the innovate or the person claiming for this product was very speculative. He was like, it may have the function. So the application had a very speculative utility. It didn't have a specific utility. Okay, so for any product or process to be patented in biotechnology, should have a specific, substantial, and credible utility. If you just speculate and say maybe future it will help in the progress of the research, no. You should have a proper utility of your product. So this invention was rejected only because this protein functionality was not known and utility was not known. Okay, so these three things like novelty, inventive step, and utility are the three filters by which an invention has to pass through to be patentable. Okay, and in different technology, the interpretations are different, and this interpretation we learn from seeing or by studying various patent case laws. So next is your excluded subject matter. Apart from these three patentable criteria, that is novelty, inventive step, and utility, there is a list pertaining to biotech in section 3 of Patent Act. Pertaining to biotech, certain points are there which is applicable in biotechnology inventions, where like if biotech invention fall in this group, then it will be excluded from being patentable. Okay, so an invention is ineligible for protection if its use or commercial exploitation is contrary to public order or morality, or causes serious prejudice to human, animal, plant life, health, or its environment. So if you are coming up with any invention which is it is prejudicial for the health of human, animal, or plant life, or which is against the public order or morality, are not patentable. Like the process of human cloning, or using the embryos, human embryo for commercial exploitation, using the germline of a human being, or using the seeds, certain seed which causes an adverse effect to the environment. So these things are like causing an imbalance to the life of plant, animal, and human, then those inventions or cannot be patentable. Next is 3C. So anything this section or this subsection of the section 3, that is 3C, mandates that discovery of any living or non-living substance occurring in the nature. Basically, anything occurring in the nature, raw, you cannot pick and go for patenting, because that is not an invention, that is a discovery. Okay, so pertaining to biotech, if we see, the extraction or isolation of any biological material is considered to be discovery. It is not a invention. You cannot go and take out a microorganism from the nature and come up come up for the patenting. It cannot be granted. Okay. Similarly, in sequence, DNA sequence or a gene sequence, you cannot as it is take it from the like animal, human or plant in its natural state to be patentable. Okay, that will come under this section only. That will be excluded under this section, stating that this invention like it is occurring in the nature. Okay, so any biological material, may it be bacteria or microorganism as a broader class, anything from the environment or from the nature, you cannot pick and go for patenting. Okay, and it should have an human intervention. If it will have a human intervention, then it is eligible. So here in the patent cases, you will see how the other party say like argues and say that this is not in discovery. I have done work here. Here is my human intervention, which makes this part or this product or process a invention, not a discovery. So here is an example. A claim was made for a novel agent for promoting cardiac development activity. Okay, and for this protein was extracted from perivitelline membrane of horseshoe crab, that is Gigas species. So from this species, from this crab, from the perivitelline membrane, a protein was extracted that was sequenced, and that was up for patenting. They have claimed for patenting, stating that utility part is it helps in the cardiac development activity. But this, during the analysis of this case, this invention or this application was rejected because the perivitelline membrane, the fluid is isolated from the perivitelline membrane, which is already in the nature. Okay, this peptide is a non-living substance which is occurring in the nature. So you cannot extract anything from the nature and go for patenting. Okay. So when this case came, when a person came for claiming, so the patent office stating this exclusion topic by selecting this subpara, they told that living or the non-living substances occurring in the nature cannot be patentable, hence your application is rejected. Next is your section 3D, which talks about new form of a known substance. You cannot come up with any product which is already there, but you are coming up with a new use or maybe a new form. Okay, without any increase in the efficacy, or there is no enhancement in the known efficacy. Okay, if any polypeptide is there in the database, which is known, which is there in the prior art, but you come up with the same polypeptide just saying, okay, it has it can exist in this form with this function, which is already known, then your invention won't be eligible for patentable. But if you come up with the same sequence with certain mutations stating that it will have an enhanced efficacy or it will have a 30-fold more functionality, then only that your application will be accepted for further examination. So here I'll just take an example. Here pre-protein A. Okay, pre-protein A is mainly is for the main purpose is for glucose metabolism. Okay, now this pre-protein A at a C peptide has some certain amino acids that is selected from a group. Okay, so here in the prior art, this polypeptide and this pre-protein with the C polypeptide was already known. Okay, mostly it was already known, and from whatever group the amino acid were selected at the C end was already known. So basically, most of the things was known, but certain interventions were there in the peptide which because here the amino acid is from separate, that is XS, which has enhanced the therapeutic efficacy of this peptide, which has enhanced the efficacy. Okay, so stating the efficacy, this application was accepted. Okay. Claim objection therapy as a result of claim, and hence the subject matter was accepted because it has a efficacy, enhanced efficacy. Next excluded matter is your 3E, which talks about mere admixture. You cannot just mix A and B stating that my product is having the function of A and B in a added form. Okay, a substance obtained by mere admixture or any process for its preparation are not patentable until and unless it shows a synergism. Synergism means it should have an enhanced or it should have an extra functionality which which the admixture is providing. Then only it can be patentable. A composition comprising known component is considered patentable only if it exhibits synergism. It means it should have a added effect. If suppose A is having a ability to if A is having some function, B is having some function, and C is having a functionality of A and B, then it won't be patentable. Synergism means A plus B gives C, which has a property of D, which means it has an extra property. So here I'll take an example. So here dormant spores of Lilicnus and Erythrobritus species fungus were taken, the spores, and a composition was formed with enzymes, fats, and growth hormones. And this composition was mainly for plant parasite nematode to control the nematodes, because nematodes were infecting the plant. To control them, this composition was used for combating the nematode. So that was the claim, that was the invention. So this composition was invention, but this application was rejected because the composition will have only the added quality of these two spores. Okay, the spores of the two fungus. So that's why the subject matter was not patentable, because here you are just adding two functionality and coming up with the product which is telling that it is two in one, the two function in one product. That cannot be accepted by Patent Act. Another is method of agriculture and horticulture. The conventional method of horticulture and agriculture cannot be patented. Like whatever the method used by our farmers and our indigenous breeders, those things cannot be taken up for patenting. So here one claim was made for growing leguminous plant as intercropping. Okay, so to improve the fertility, like in one season they will grow a leguminous plant, in the next season they will grow non-leguminous. So it is alternate or intercropping was this method was claimed, come up for the claim, because the utility is because it improves the fertility of the soil by augmenting or by increasing the nitrogen content of the soil. So is this subject matter will be patentable? No, because the intercropping leguminous plant is a very common practice which is done by our farmers. Okay, so since it is an agricultural and horticultural method which is owned by by farmer and which is a conventional method, then this is not be patentable. So that's why this invention was rejected, or this application was rejected to be patented. Next is any process for medical, surgical, curative, prophylactic, diagnostic, therapeutic, or any treatment basically any treatment for curing the disease or making or any animal rendering free from disease or increasing the economic value of that product is not patentable. Okay, so why it is? Because you cannot protect a process of curing a person or making that person free from disease. It should not be monopolized. It should be for the general public. The process of all the treatment should be for available for the general public. So that's why this treatment methods, different treatment methods are not eligible for patent. Okay. Example, if suppose you have a method of treating a sheep to increase the wool yield, or the method of artificially inducing the body mass of the poultry is not patentable. If you say, you can say, okay, I'm coming up with this invention to increase the wool yield, to have an utility. But here you are, if you see the previous thing, to increase the economic value, you are increasing the economic value just to get an wool, you are exploiting the sheep. Okay, so application of and this application is not applicable to a cosmetic purposes, for any for massage, for hair treatment, or for the lift up, or any cosmetic things where you are not, the treatment is not there to treat any human or animal, then it can be patentable, because cosmetic process is not comes under any surgical medical process. Okay, so even the prosthetics, manufacture of prosthetics, artificial limbs, these can be patentable. Okay, plant and animal in a whole, as a whole, plant and animal cannot be patentable, but a microorganism can be. Okay, or any part of plant and animal like seeds, varieties, or species cannot be patentable. And even the essential biological processes are not patentable subject matter. So here, a method of producing at least one substantial pure hybrid seed, that is plant and crop, comprising a step of. So here, if you are producing a hybrid seed, you have a steps like producing a male parent from a male fertile, breeding the male parent with the male parent, female parent, which is substantially male sterile, harvesting the seed from the female parent, and then containing the pure seed. So the claim involves the step of crossbreeding for producing pure hybrid plant and crop. So it is, and all the methods here used are essentially biological methods, which is already there, or in the either in the conventional way, or it is there in the nature. So these methods are not. So that's why this invention coming up with the using the normal essential biological process won't be patentable. Okay. So mathematical or business method or a computer program per se or algorithm cannot be patentable. Pertaining to, if we see in biotechnology field, the bioinformatics, anything related to coming with an algorithm which can read the sequence and read the particular polypeptide sequence or any sequence, those things, the algorithm may not be patented as per this section 3K. 3P. This talks about anything which is present in the traditional knowledge, or in or any aggregation, duplication of any known substance which is there in the traditional knowledge, which is there with our ancestors, which there in the prior art, will not be a patentable subject matter. Example, one claim was done by an application for a serum of a pigeon possessing an anti-paralytic activity. Okay, so that was so he extracted the serum from a pigeon stating the utility that it has anti-paralytic activity. But when this application was analyzed, it was known that it was already there in our traditional knowledge that pigeon serum was used for treating paralysis. It was there a publication in the journal which discloses the use of pigeon blood for treating paralysis. Okay, so it was already there in the traditional knowledge as pigeon serum to be used for anti-paralysis. So that information cannot be picked up or taken up for patenting. Okay, so these are various exclusion topics, or these are the excluded subject matter which prevents a patent to be for application to be patented. Okay, so for any invention to be patented, it has to first cross through novelty, inventive step, and utility test, and it should be excluded from this listed excluded subject matter. Okay. Now, next is Budapest Treaty. So, like as per Indian Patent Act, and this Budapest Treaty is like you have to comply with the international forum. So, in the as per Paris Convention, all the countries should sign this treaty who are in the WIPO Paris Convention members. So here pertaining to biotechnology, whatever invention you are coming up with stating a microorganism, you have to submit that microorganism or sample of that microorganism to IDA, that is your depository, International Depository Authority. Okay. So broadly, if you are working on any microorganism and you are seeking a patent to it, so that microorganism you have to submit at an International Depository Authority, which is in India, which is at Chandigarh and Pune. Okay, and this is applicable to all the country who are signatory to the Budapest Treaty. So this is the prerequisite. In India, it is a prerequisite as per Budapest Treaty to submit your microbial sample to the International Depository Authority. Another compliance for a biotechnological applicant to make is to, as per Biological Diversity Act, you have to acquaint or you have to let the body, as per the Biodiversity Act, to that we are using the microorganism from the environment, okay, or from this origin or from this geographical area. Okay, so you have to acquaint before you start your research. You have to know, you have to know the National Biological Diversity Authority that I am using this resource from the nature to perform my research, because your research will fetch you it is having a commercial value. So it should not be commercially, the resource should not be commercially used, or it should be used in a regulated way. For that, you have to acquaint or you have to inform the authority that we are using the this microorganism from this biological origin. Okay, so till now I have discussed all the excluded subject matter in the Patent Act, which tells whether a patent is or whether an invention is patentable or not, and the filters, 3 filters which they have to pass, that it has to pass novelty, non-obviousness, and utility filters. Okay, now I will just take you to the journey through the of biotech patent evolution, like how it evolved. So if we see in 70s, it was the time when there was the growth of biotechnological process like recombinant DNA technology, genomics, proteomics, gene therapy. all these things were booming. Okay, so there was a lot of research also, there was a lot of research at genetic level were going. And since we know why we need patent is required, so that there is more investment in R&D. Whichever technology, wherever there is more patenting, or where in whichever jurisdiction where there is more patent or there is more patenting culture, it means the people or the government is investing more money in research and development. Okay, so if we see US law say invention means, from their term, any process, machine, manufacture or composition of matter, or any improvement thereof, anything, it surpassed to all the technology, even to biotechnology, like whatever comes under this will be patentable. Okay, so biotechnology comes under the composition of matter or manufacture. Okay, so in the patent cases in the US, if we see, they use these lines to prove that your like work is an invention or a patentable invention. Excluded, what all things are excluded subject matter in US? They are any laws of nature, physical phenomena or abstract idea. So that is like it is discovery, it is already there in the nature. Okay, like gravity, gravitational force, all those things, physical phenomena which is there in the nature, those things you cannot come up for a patenting. So as I told, in 70s there were a lot of work on microorganism, and US was not clear, or not even US, in the worldwide, the patenting in microorganism or living thing, it was not clear. It was very vague. But since the cases came, since the innovation and application came up, then the flexibility in the thought process or in the flexibility in the filters were came up. Okay, so here, first example, first one case was between Funk brothers and callow inoculants. Okay, so here the Funk brother came up with the consortia of microorganism, or a group of microorganism, or a mixture of a culture of various microbacteria, which helps in nitrogen fixation in leguminous plant. Okay, so the claim was that these are non-inhibitory. There are 5 bacteria and each were not inhibiting each other, but they are performing function independently, and this culture can be used in crop to fix nitrogen. Okay, to fix nitrogen. But when the analysis was done by the court and by the patent office, when the analysis was done, it came to know that this non-inhibitive strain, or the ability of the function of all the bacteria and non-inhibitiveness, it is not a human intervention. The person has just taken up 5 bacteria, they have mixed it in a culture, and they have come for patenting. That will not be eligible for patent, because it will come under the nature law. That means it is already present in the nature, it is already occurring in the nature. So that was the decision by the court, stating that since you are coming up with the organism which is already there in the nature, we cannot grant patent for this. But on a contrary, like any decision in whichever jurisdiction, even if you see in US, India, Europe, any decision is not closed door decision. Here they told naturally occurring substance or living organisms are not patented. But during the time when many cases came and they proved like how it is different from occurring in the nature, and then they got the patent. Okay, so it means how you combat or how you justify your invention also. So here is one very good example. So a patent application was filed for purified vitamin B12 isolated from fermented material. Okay, so court initially told कि B12 it is already there in the fermented material. and it is already there in the nature. So there's no human intervention. But court, the court told it is a, initially they told it's a doctrine of product of nature. It is a product of nature, so we cannot grant a patent for it. But finally they told that patent may be granted to product of nature provided, now they have put an but into it, it is new and useful composition of matter. Okay, taking out, extracting B12 from the fermented material, it takes a like creative mind, a creative knowledge to come up with. It is present in the nature, but you cannot use that B12 as you use the purified or extracted B12. Okay, and so that's why here the applicant conveyed that yes, it is a product of nature, but the human intervention is extraction, and extracting and making a new and useful composition of matter. The B12 which is present in the nature was not as useful as my product B12 because it is a pure isolated and purified. So with this argument, the patent was granted, and that's what the court became little flexible. They told patent may be granted to product of nature provided it is new and useful composition of matter. Okay, so here is the intervention that he extracted B12. Now another landmark case in US in the biotechnology is your Diamond and Chakrabarti case. So what happened in this case? So till now in US they told microorganism or living things are not patentable. Okay. So here Anand Chakrabarti came up with a genetically modified Pseudomonas bacteria. Okay. This is genetically modified Pseudomonas bacteria in which he injected or he through recombinant DNA technology, he transmitted three different genes who are capable of degrading oil spills. Okay, during those days, oils were transferred from one continent to another continent via ships, and somewhere accident used to happen. Oil spills on the ocean, or even industrial waste. So oil spills was the major environmental issue. So Anand Chakrabarti came up with the Pseudomonas bacteria and injecting 4 different plasmids. These are from different plasmid which are capable of degrading 4 different oil spills, different type of oil spills, and for that gene are transferred to this Pseudomonas bacteria. Okay, so on appeal, the US Supreme Court held that invention is patentable because it's a new bacteria. This bacteria does not found in the nature. This bacteria is not the product of the nature. Here the intervention is through the recombinant technology. This person has introduced the gene for degrading the oil spill. So here hence the product of the nature, and here as is patentable provided the human intervention is there. It means here genes have been introduced to it. So this was the first case where a microorganism, man-made microorganism was patented, and it opened the door for numerous application for in which a new product or new product or process have been used by have been come up by using recombinant technology. Okay, so after this, even transgenic animals like genetically engineered oysters were patented. Okay, then even Howard mouse in 1988. This Anand Chakrabarti case was in 1980s. In 1988, the mouse that was known as Howard mouse was also patented. Here, the mouse was it was genetically engineered to have a cancer. There was disruption of the cancer, and they used to do the drug testing on the cancerous mouse. So this model, the cancer model, the mouse was patented because the this case opened the door for genetically modified or microorganism, plants and animals. Another case which happened in 2014 recently, which told that is molecular pathology and genetics. In this, they told that naturally occurring gene. Again, the US was going mostly towards naturally occurring things in the nature. They were just little bit manipulation and they were coming up with. Now, this case again closed the door and told that again genes cannot be patented. So in this case, Madrid scientists came up with two genes, that is BRCA1 and BRCA2. These are mainly responsible, or the mutation in these two genes are responsible for breast cancer in women. Okay, so they got the patent prior, but so all the testing and everything was concentrated with this Madriad because they had the monopoly to use this invention, and various other companies and NGOs filed a case against the Madriad stating that these two genes, these are naturally occurring genes. And since recombinant DNA technology by like this became very obvious at that time because using gene therapy and extracting gene, then you're amplifying the gene through PCR. They were very like handy tool to do the gene. So that's why they told it is very obvious. These two genes are already patent, like it's already there in the nature. And they once they patent the gene, it is not available to all the people because at that time they were charging $1,000 for one test. But various NGOs and the companies wanted these tests to be available to the common people. So they told it cannot be patentable because it is there in every human body. So with this argument, Madriad genetics patent was declined and it was rejected. Okay, because the BRCA1 and BRCA2 genes are already there in the human body. Now, with this much of development in US, stating prior it was that no naturally occurring organism or anything which is there in the nature cannot be patented. Then with the various case laws, the law became little flexible with certain conditions attached to it. So in India, at that time, many like it was even they wanted, even in India, Indian patent law and in Indian researchers society or Indian biotech fraternity wanted a broadening in our statute, that is Patent Act, so that we can include our various things which can be patented. So whatever excluded subject matter I discussed before, they had been elaborated with the amendment in 2002 and 2005, and the patentability criteria have been more clarified and elaborated in India too. Now, even in India, now you can go for microbial or microorganism patenting, or even for a gene patent if it is a construct, not a naturally occurring gene. You cannot, naturally occurring gene is not patentable, but if you are constructing with a promoter or a terminator and if you have a construct, then it is eligible for patenting. Okay. So natural occurrence of microbes. Here also natural occurrence of microbes are not patentable if you are doing a certain genetic engineering and providing an utility, and that recombinant DNA, recombinant microbes is having certain utility, then it is patentable. Induced mutation. If you are inducing a bacteria to perform a particular function, then it can be patentable. Consortia of microorganism. If it is having a synergism, like if you have a culture having 100 microorganism, but providing a functionality, that functionality should not be just addition of all the function of the bacteria, the performing function. It should be something more. As I told you, it should have some synergism. It should have a plus or enhancement. Then only it can be patentable. So gene in the form of gene cassette, which is performing a particular gene phenotype, then only it can be patentable. You cannot per se take out a gene from a human body or animal or a microorganism and go for patenting in Indian scenario. Synthetic gene, which are nonsense, which are not forming any useful protein, or more precisely, if we say it is not having us utility, then those genes also cannot be patented. Okay, promoters can be patented. Okay, and transgenic and animal and transgenic plants, these two are like statutorily they have not told it is not patented, but the doors are open. Any case has not came for transgenic plant and animal in Indian scenario. Okay, non-patentable invention in Indian scenario as gene occurring in the nature, nonsense mutation, any plant and animal except microorganism. Any plant and animal is not patented in Indian scenario except microorganism. Microorganism can either it, and it is a very broad term. Microorganism includes bacteria, prokaryotes, fungi, yeast, viruses. Okay, so it all it surpasses everything. Now, one case in India set in a landmark for patenting a live living organ, a live microbes. Okay, so this was in Calcutta High Court. So here was Domineco. Okay, so Domineco is a US company who came up with a vaccine which is bursitis vaccine. Okay, this bursitis vaccine was mainly for the poultry or hens to combat the bursitis infection. Okay, so basically to combat bursitis infection in poultry animals, bursitis vaccine was came up. Okay, so initially they told, and this bursitis vaccine had attenuated virus. Okay, attenuated virus. It means it had a living substance. So initially the patent office told that invention must be new and it should be useful and manner of manufacture. So initially they told ki it is a living things, whether we should, so it was in a dicey situation whether a patent should be granted or not. Okay, so but court stated that the end product or the whatever vaccine we have, it is a vendable product which has a substance, which is a process of manufacture. It is manufactured, it is through recombinant technology, it has been manufactured to fight against the bursitis infection. Okay, so so it is here also through recombinant technology, the organism was used to fight against the infection. So it has a utility, it has a human intervention. So these argument won, and this patent was granted, or this patent was held, whether it was validated. Okay, so with this global development in the gene patenting and the gene construct patenting, India also became liberal in patenting in the biotechnology field. Okay. Now, if we see the EPO or the European Patent Office, so little bit different. In Europe, they say that they are for them natural occurring organism or natural occurring living things can be patentable. Okay, so there it is a little bit different. So subject matter of an invention is a sequence or a partial sequence of a gene, the structure of which is identical to the structure of natural sequence or partial sequence of a human gene or thereof. So Europe culture is different. So there they told that any natural gene, occurring gene or microorganism is patentable until and unless that person or the applicant comes up with a utility, with an obviousness, like non-obviousness, and it should be new. It should not be known to the in a prior art. But if it is occurring in the nature, it is patentable. So here EPO is different. Europe culture is different. Okay. So, and one of the essential condition for the grant of patent for a genetic sequence is that it should have an industrial applicability, and they emphasize more on the industrial applicability part in Europe. Okay. So here I'll take a case law. So a recombinant DNA molecule encoding a polypeptide for an amino acid sequence, that is a polypeptide, is responsible for at a locus, responsible for aluminum tolerance in sorghum bicolor. Okay, so there was a tolerance, and this was performed by extracting a gene which is already there, in the nature, and that was patented in Europe. So in Europe culture, if it is naturally occurring, you are isolating it, coming up with a utility or with some functionality, then it can be patentable. Okay. Now, in if you see the utility part, so function of a protein can be seen at different level. How will you decide the functionality? You will see functionality at a molecular level, cellular level, and a biological level as a whole. Okay. In Europe, one case came for neutropene alpha neutropene, which is a protein belonging to tumor necrosis factor. Okay, this is a ligand superfamily. So it came up with the invention, but their utility was very speculative. They were not they were not very much sure about the utility part. So the application was rejected in European board, stating that it does not have a utility. Okay. Next is, I told you all the essential biological process. Essential biological process are excluded from patent. Like what are the essential biological process? Like your crossing over, selection, selection of crop. So all these things which is common or which is there in the nature or which is used by the farmers, so those are not patentable until and unless there is a human intervention which determines whether it is a discovery or invention. Okay. So in a case, in a Lubrizol case, what happened? A high quality hybrid was produced. So to determine its patentability, consideration is made to essence of the invention, looking into the essential biological process, human intervention, and impact on the result. Okay, so it involves to get a high quality hybrid, it involves selection, crossing of the parent plant, evaluation, hybrid, and then selecting a suitable one. So here the applicant is using the process which is there in essential or which are the essential biological process, but human intervention is they are selecting it, then whichever is the best one, they are crossing the best. So here the human intervention prevails. So stating this, that human intervention is using the biological process in a systematic way which is not there in the prior art, then it is eligible for patenting. So basically any excluded matter or any filter is not stiff or it is not a closed door. By your argument, you open the door or you make a precedence in the field, and you can have a various further application equals to that. Okay, so another issue in biological invention is the moral issue. Morality, as I told you, one of the excluded is morality. It should not be contrary to morality and ethical issues. Like you cannot have a transgenic human or transgenic animal by slaughtering or using the embryos and commercially. Okay, so here moral issue comes wherever it crosses the morality, then that product or process cannot be patentable. But still, that issue, this morality issue, is open for discussion. Okay, it is not a closed door. Okay, in Europe, a pharmaceutical company, Upjohn, came up with a mouse in which hair growth gene has been disrupted so that hair loss was there. So basically they were creating a model of hair loss and then they were testing the drugs on it. So this in EPO was rejected. In Europe, the ground on morality, rejection of a patent on morality is more. Okay, so there they told that the invention was immoral and hence it was not patentable. But
Account.
in contrary,
I've
okay,
already
here,
decided to close the account, okay, which I had opened
okay,
last
so
March,
here,
that is 18,
it's hello.
March, okay, and I
Okay.
just wanted to close the account. I think I have already given
Hello,
letter also.
I can
So
hear some noise, ma'am.
Ma'am, I've muted the participant.
Okay, 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 okay. So here, disrupting a gene in a mouse, it was a morality issue. That's why that patent was rejected. But this is not a closed door. Okay. In this contrary to it, another case was relaxin case. So what happened in this case? The relaxin was extracted from the human body during the childbirth. Okay, so many group told that it is against the morality because you are using the pregnancy to extract a product. Okay, but the argument was it was a voluntary action. If you want to donate your relaxin or not, it was a voluntary action provided to the pregnant ladies. So there was no contrary or there was no moral issue because it was through the consensus that they were extracting the relaxin from the pregnant woman during the childbirth. Okay, so here there was a no moral ground. Initially, the morality was stood stating that they are using a human body to extract, but it was a consensual thing. So during the with the discussion, they hold that this can be patentable because it is so here the morality was like they told it is not against the human dignity. It was proved through the argument and the patent was given to the relaxant. Okay, so still the topic is open whether animal as a bioreactor, like can I inject a gene to a cow and get particular protein? So it is open for discussion. The on the ground of morality is not closed. Okay, so even for a human being to include human embryos, to study embryonic tissues, and to include germline therapy, to have a human disease model to test drug. So these are subject to open for discussion on morality issue. Okay, so this is a chart which says in different jurisdiction like India, Europe and US, what is the status? Like if you see gene isolated or DNA sequence, which is complete no in India and US. In India and US, the occurring genes cannot be patented, but Europe you can have. Okay. Similarly, if you see others are cDNA sequence is patent in all the jurisdiction, Europe, India, and US. Like cDNA is not naturally occurring. From mRNA, you are removing the introns on and then making the DNA. So cDNA can be patented. Okay. Now, altered mutation or variant of a gene or DNA sequence, this is patentable in all the jurisdiction. Okay, primers, probes, it is present in all the jurisdiction, but in US, certain cases are there where the patent has been rejected because of certain natural issue, like it is present in already in the nature. Okay, so kits, this is present like diagnostic kit and primers, probes. These all are patented in the all jurisdiction. Okay, isolated protein sequence. Okay, it is not patented in India, but Europe it is, and in US in some cases, yes. Okay, so this, due to patent cases, precedence has been set and opened the door for further application. Okay, so gene patents, composing pharmaceutical, biotech, agriculture, bioindustry, but it gives, must be a patentable to give firm a confidence to invest money. Why we need patent on these fields? Because this gives investors to invest money, even governments to take up and put the funds into, so that we can have a gene-based drug and genetically modified crop. So if we have more money in the R&D, we can come up with the various drugs and genetically modified crops. Okay, and these various filters like novelty, inventive step, utility, these can be argued. Okay, this conceptual issue in patenting biotech invention problem can be combated by argument on these topics. Okay, so here I conclude my talk. So thank you. Any questions, please?
Participants, if you have any queries, please unmute yourself or post it in the chat box. Ma'am, we have a message from Dr. Raki, very informative session.
Okay.
Anyone, any other participants?
I know who is speaking. I know who is speaking.
Ma'am, I think that's all the questions. I think participants didn't have any queries. So, ma'am, I, on behalf of the Department of Biotechnology, the Oxford College of Engineering, we would like to extend our heartfelt gratitude for this detailed information about the patenting laws, restrictions, all these details. Thank you so much, ma'am, for this wonderful lecture.
Thank you so much. Thank you, everyone.
Still on sentence performed.